Registrar Nizer Bhuneshwar. Thank you, sir. It is my privilege to welcome Mr. Mohammad Shakil, a distinguished former registrar of IIT Kanpur and IIT Goa, to grace this event. His outstanding contribution to academic administration have significantly shaped the operational excellence of these two Indian premier institutions. His visionary leadership and ability to manage complex academic ecosystems have left a profound impact, ensuring a seamless functioning of these institutions while fostering innovation and growth. Mr. Shakti's dedication to excellence and his contribution to development serve as a source of inspiration for all of us. We are truly fortunate to have you, sir. Please join me in extending a warm welcome to him. Sir.
Thank you and very good afternoon to everyone. In fact, for last 2 days I am traveling. So when I came here, then when I was coming here, so Dr. Naik, my very old friend, a very close friend, he said, "Come and spend some time with us with this institute." And I'm really impressed to see the infrastructure and all these things here. I served three institutions, not I. I served IIT Kanpur for about 17 years. After that, I joined IIT Mandi also in Himachal Pradesh. I spent about 3 years there, and then for 5 years comes in IIT Goa. Recently, I retired from IIT Goa, so I'm a free man, just delivering talks to different. I'm associated with different institutions. Some talks on academic administration and some motivational talks. So when I joined, just I want to share a few things here because I know many speakers are waiting. We are already late here. So when I joined, I was in CAG before coming to IIT. So from bureaucrat to IT academic administration, there's a long journey I performed. So when I joined IIT, so I asked, like, there are many IT people, they know the vision of IT. I said, what is the difference of IT and other institutions? Why the IT is a brand? Why other institutions are not a brand? And they told me, see, we are not producing job seekers. We are producing job creators. So that is the difference, in fact, in ITs and other institutions. And that time, and sometime in 1980s and 90s, when this incubation started, this things were started. Then IT Kanpur established this SIDBI Incubation and Innovation Center. Then I interacted with the faculty members there, in fact, to know, in fact, how these cells are functioning and what is the purpose of these. So they said the idea is establishing this innovation center is to stop the brain drain of the country, in fact, because most of the IT students, they studied here and leave the country, in fact. So government of India spends a huge amount on these students. But when they became after graduation, they left. They leave the country and they serve US and Europe and other countries. So they are making other countries rich. And but we deserve for that, in fact. So they said, no, we want to do this because they should work here. They should come with the innovative ideas, develop a prototype, start startups, and give jobs to others. So this is the purpose of IT for education. And I'm happy here, in fact, and it depends on visionary leadership also, because I saw this Dr. Pati's this establishment and all these things. Private universities are not there in picture. In fact, very few private universities are there, in fact, who are creating these cells here. I also got opportunity to visit many universities abroad, particularly in the US. So what I found, everybody you also noticed that most private in US, if you see the most private universities are top universities, MIT, Stanford, and Yale. But in India, the situation is reverse. Here, private universities are, not, as developed as the government institutions like ITs and ITs. The reason is they have funding things. They have. It's not funding only. It's also a vision. If you're a visionary person, definitely institution will develop. You will generate funds. I know IIT Kanpur and other IITs, they have huge corpus. They are not dependent on government funding alone. They have their own corpus, 200. I know when I was in IIT Kanpur, they have a corpus of 250 crores. And you can imagine the interest earning on this corpus. And every time they are, every year, their alumni, they are contributing huge amount. Recently, 100 crores was given by one Mr. Gangawal, who's a co-founder of Indigo, for establishing a scientific this institute in IIT Kanpur. So why I mean ITNs are giving funds to ITA? Why the other institutions are lacking funds? The reason is because we are producing here only job seekers. We are not producing job givers. And I'm thankful to Professor Wadhi because he has taken this initiative. He is really a visionary man. He has established this center here. And through this center, I would request, I would encourage the students and the faculty member here to come forward with new ideas, develop prototypes, I mean, develop some startups and do something for the country. And that is this way you are not serving your institute or you are also serving your country. Because if you see the Government of India initiatives, Atmanirbhar Bharat, Make in India, all these initiatives are based on these entrepreneurship. So with these words, I think I'm thankful to everyone for coming here and for this event, for this conference. Thank you very much.
Thank you, Mr. Shakil, for your kind address and invaluable insights. I'd request Dr. Himanshu Mohan Padi, Chairman, Sophitorium Group of Institutions, to please present the memento to Mr. Shakil on behalf of Nasi, INSA, and also Sophitorium. Register. Thank, thank you, Dr. Padi. It's indeed a very thoughtful gift for the dignitaries. I'd like to express my heartfelt thanks to the esteemed delegates on the dais for their valuable contribution. Your insights, expertise, and reflections have greatly enriched our discussions and have perfectly set the stage for some engaging talks by our distinguished speakers. We are grateful for your time and support and commit to sharing your knowledge with us. Dear delegates and researchers, it is now time that we witness our experts share their profound experience in science and impact and its impact on humans. I'd like to request our first speaker, Dr. Soma Chattopadhyay, an esteemed scientist at the Institute of Life Sciences, Bhubaneswar, to please proceed with the with her talk. Before that, I'll just give a brief introduction about her. Her groundbreaking research spans over 2 decades, focusing on virology and immunology. With a notable contribution in development of sensitive antibodies against the chikungunya virus, which have significantly advanced our understanding of viral infections and host pathogen interaction. Recognized with prestigious awards, Dr. Chattopadhyay continues to inspire the scientific community with her dedication and excellence. We are privileged to have her with us today, sharing her insights and expertise that undoubtedly will enrich our sessions. I request all the speakers to um, I would request all the speakers to restrict the talk to 50 to 20 minutes due to time constraints. The stage is yours, ma'am.
Sure, I'll do it. Put it down. I'll do it. Very good afternoon to all of you. I'm thankful to Professor Kundu and Dr. Chatterjee and definitely Dr. Pandi for inviting me for this conference. Science for Society. I can see there is a question mark in Dr. Naik's face that what is FIBS? So I'll just explain this. So I am a fellow of National Academy of Science, Allahabad. So that is why FNASC. And then FIBS, I'm fellow of Indian Virological Society. So we have a society, those who are working on virology. So for my contribution in medical virology, I'm a member, fellow of that also society. Uh, so I have given this topic how to control emerging and re-emerging viral diseases, infections. And I think for last 3-4 years, everybody has seen the power of viruses, right? The smallest creature, if I say it's a creature, because we know in textbook it says it's between living and non-living. So, but this has made the whole world into a stop. We literally have never experienced this kind of pandemic before. And yeah, in past there were few other pandemics, but this was really a huge pandemic where with this our modern technology and all these things, still we suffered a lot. So today I'll be talking about a virus which is chikungunya virus. But this chikungunya virus belongs to a RNA virus. And when we talk about viruses, that we know that RNA viruses are more dangerous and deadly and difficult to control. Even for our COVID, we have seen that within these 4 years, there are so many different variants have appeared, and we had to understand differently regarding all these virus strains. So I'll give little overview about this chikungunya virus, and then in our lab we are working on different antiviral drug development. That can be through repurposing of different existing drug, or we are also synthesizing new molecules to understand their antiviral property. And there are also another way that we can control viral infections when we can understand the host pathways which are modulated by the virus for its own replication and infection in the host system. So, just to mention that this chikungunya virus belongs to this alpha virus genus. In that genus, especially these viruses are of RNA as a, with their genetic material, and the genome size is very small. You can see that it is only 12 kb long. And this is like our mRNA of the host system, where there is a 5 prime cap and 3 prime poly A tail. So if you can see below that the viral genome is having a non-structural protein coding region and a structural protein coding region. And the whole replication and protein expression, everything takes place in the cytoplasm of the host cell. And this virus is mainly transmitted by mosquitoes. These are Aedes aegypti and albopictus. And the symptoms are sudden onset of high fever, rash, nausea, vomiting, most importantly, myalgia and polyarthralgia. So these are basically pain. If you have experienced chikungunya virus, or if you have seen anyone suffering, you will understand that they always complain about the pain in the joints. It is very difficult to, do any small work. One of my student, who was by that time 25, 26 years old, and she got chikungunya infection. Even after 6 months, he was not able to comb her hair. It was so difficult to turn. I mean, so often it becomes very painful and difficult to do day to day activities. And this pain usually persists 6 months, even 1 year after the clearance of the virus, also in some cases. So, as I mentioned, that the viral genome is having these non-structural proteins, there are only 4 non-structural proteins which are required for the viral replication inside the host system. So if we see, these are different function of the proteins, which I'm not going into details. But when we try to understand how the virus can be modulated after, so that we can control the disease, we need to understand the function of these non-structural proteins. So similarly, there are a lot of other host proteins which are required for efficient viral infection, starting from attachment to the host cells through a receptor, and after that uncoating of the genome, then synthesis of the viral protein, transcription, translation, and again packaging of the virus and egress from the host system. All these steps require lot of host proteins involvement. And if we want to really understand the viral biology and to develop any molecule to interfere into any of these stages, we need to understand what are the host factors which are making this virus efficiently replicating inside the host system. So our lab actually works on different aspects of this virus infection, where we develop different reagents. Because when I started this my lab and working on chickpea, the in short we write chickpea, so we did not have any reagents to even work. So I developed all the clones, all the antibodies, and currently I'm just happy to say that we are actually selling this non-structural protein antibodies through a biotech company. So this is there in our their catalog, and we have already sold around 14 countries. These 81 receptors are important for chikungunya virus infection. So these 81 receptors goes up after viral infection. So we were trying to understand that can we really see whether it can modulate chikungunya infection. So we have done the repurposing of this molecule. So why we have taken telmisartan? Because as I mentioned, this is already a FDA approved drug. It is, people are using this for hypertension. I mean controlling hypertension. And what we have seen that when we look into its binding ability to the viral protein, but through the in silico analysis, we could see that it is having affinity for binding to the viral protein, which can give you some confidence that it may interfere with the function of those enzymes. And then this was also seen in few other viral infections before. So we took it. First, we have done a in vitro analysis, seeing the toxicity of the drug by MTT assay, and then we have performed in vitro antiviral assay, where you can see we have used vero cells or macrophage cell line, which is a raw cells, and then we have infected the cells with the virus, added telmisartan, and then analyzed through different assays like confocal, Western blot, FACS, and then also plaque assay, which is a gold standard method to see, which you can see this violet color, you can see the white color plaques, and in presence of the drug, you can see the reduction of these plaques. And in this confocal, it is very clear, if you can follow this, that E2 protein is actually this red color protein you can see. And then in DMSO panel, the second panel, you can see that this is the amount of viral infection and expression of E2 protein, which is reduced significantly in presence of telmisartan. So this indicates that it is having antiviral activity. We did lot of analysis because of the time constraint. I am cutting this story short. And then after seeing this efficacy in the in vitro cell culture system, we have taken it to the mice model, and where we have used C57BL6 mice. These are 10 to 12 days old mice, and we have given viral infection. 3 to 4 hours after infection, we have administered the drug, and then every day one dose of the drug which has been given. After 5 days post infection, usually what happens, the mice which are injected with chikungunya virus, they do die. So we have a nice in vivo system, and that is why we have followed them up to 5 days post infection after giving the drug. So you can see here, after chikungunya infection, they develop hind leg paralysis, as probably you can see in the first figure. Hind leg paralysis, then both the leg paralysis, and they eventually die. Whereas in presence of telmisartan, we can see that they are not developing any paralysis and they are able to walk properly. And if you can see the viral titer from the serum and from other tissues, we can see reduction of the viral protein in different tissues as well as in serum, indicating that it can actually reduce viral infection in mice. Also, we could see reduction in inflammation. So if you can see these tissues, which we can see are damaged after chikungunya infection in the muscles, and also infiltration of immune cells, which are really restored when telmisartan is administered. And a graph showing clinical score, and based on the scoring that we have given, the clinical score is very high with chikungunya infection, and it is reduced in the graph. And when we did a survival curve analysis, you can see that all the mice died after 4 days post infection, whereas in presence of telmisartan, we can give them full survival. So after that, we have taken the human PBMC from 3 different donors and performed the same antiviral assay. And we could see that in presence of telmisartan, the viral load goes down. Yeah. So this is just a schematic representation how this telmisartan is working. I am not showing all the pathways analysis that we have done, but what we are seeing that telmisartan can actually work to reduce the inflammation and the injury which is happening to the muscles by actually two ways. One is that by reducing the AT1 receptor on the surface of the cells, and also one work from Delhi IIT, they have published showing that NSP2 protein protease activity is inhibited in presence of telmisartan. So together, by targeting the viral enzymes as well as the host pathway, telmisartan can protect, basically give reduction in viral infection in host. So I'll just quickly touch another story, which is a molecule MBZM-NIBT. It's a novel small molecule which has been synthesized by our collaborator Dr. Subhuti in Shoa University. So he's a pharmaceutical chemist, and we have a really long collaboration with him, and he does synthesize different molecules with antiviral property, and then we can test. So this is a molecule which we have patented, and you can see that this 2-methyl benzimidazole was one of the component which was known before for vaccinia viral infection, and it was taken from the market due to a few difficulties that people were facing. And IBT is another molecule which is also part of different antiviral molecules. So what he has done, he has conjugated these two molecules, and we got this MBZM and IBT. And with this molecule also, we have done in silico analysis against different viral enzymes to see whether this hybrid molecule can give better binding affinity towards the viral enzymes. So you can see with NSP1234, we could see better binding affinities than its other decoys. So after this in silico analysis, we went on to do the wet lab experiment where when we saw that this molecule is having more affinity towards the C terminus of NSP2, which contains protease activity. So we have cloned that, expressed and purified, and we looked into for its binding affinity, analyzed this by fluorescence, and we could see that this MBZM can bind with the C terminus domain of NSP2. I'm not going into details. Also, we have used a substrate to see whether this substrate, this protease is cleaving this substrate, and which can be reduced in presence of MBZM. So this is also a analysis in the confocal microscope. You can see that E2 protein expression is reduced in presence of MBZM. So this being a new molecule, we had no idea what this is targeting at, and we did a pre, during, and post different conditions. So what we do usually, we treat the cells with the drug and then give viral infection. Or in the during condition, we pre-incubate the drug with the virus and then see whether that is reducing infection. And the post condition, we allow the virus to infect the cells and then we add the drug to understand that what is the mechanism, how this drug is interfering with viral life cycle. So here you can see that we would see good amount of in the pre-condition, we actually did not see any reduction. In during also, we did not see any reduction in viral infection, whereas in the post condition, this one, you can see that we are able to reduce the viral infection. So then in order to understand during post infection what is the actual phase which is getting inhibited in presence of the molecule, so we do a time of addition experiment here where we add the molecule at different hours of post infection. We allow the infection to go for certain time and then add the drug. So here we can see this molecule can actually control the viral infection for a long time. So, which gives an indication, even at the last point, if you can see at 7 hours post infection, we are able to reduce viral infection. Whereas ribavirin, the red bar, is actually a known antiviral molecule we have used as a control, and we could see that this molecule is even working better than ribavirin. These are different cytokines we have seen to see that the pro-inflammatory cytokines are reduced in presence of this molecule, because inflammation is one of the important problem, or it is a parameter after chikungunya virus infection which also need to be controlled. So here we have done the toxicity level study in the animal before we can take it into the animal model study. I'm not going into details, but we could see that this was really a safe molecule. Also, we have done chronic toxicity testing. So, based on that, we have taken this 15 mg per kg dose. And can I show this video? Video can be, I think, or I think, yes. So, if you can see this, you can identify the one which are Chigvi infected and the which are with drug treated. So you can see both the legs are proper. That means this is the one which is drug treated. And you can see the other groups. They are walking very properly. They are with the drug treatment. And these two, you can see that one leg is sticking out. So single leg paralysis happened. After one day, we will be able to see both the legs are paralyzed, and they will have difficulty. They will lose the weight, movement difficulties, and then finally they all die. And we can actually protect these animals with MBZM. Yes, so here you can see the level of viral protein after MBZM administration, and we can see in all the tissues we have seen muscle, spleen, kidney, liver, and brain. We can see reduction in viral level, and the inflammation that we see after chikungunya infection, that is also reduced in presence of MBZM. And also we do see splenomegaly in the spleen, enlarged spleen, which is also reduced. And in muscles, after viral infection that I was mentioning, that there usually muscle damage happened and infiltration of immune cells, that is also resolved after administration of this drug. So this is just to show that we can give protection to the mice in the survival curve. So when we gave the drug 24 hours interval, we still saw that there are few mice were dying. So we have increased the dose every 12 hours, and then we can give full protection to the mice. So this is the work which we have done in collaboration with NICER. So Dr. Shubhashish is here, and they have collected the human PBMC from 3 donors. And in case of human PBMC also, we can see that great reduction of the virus. So basically, we are now finding what are the host pathways which are modulated in presence of this molecule through our RNA-seq and proteomics analysis. But as of now, what we have seen that this can work on the protease activity of NSP2. It binds to the protein and then reduce its protease activity, which then interferes into the replication of this virus inside the host cell. I will not go into details, but as I mentioned that in order to control the viral infection in the host cell, we need to also identify different host proteins. So I'll just mention the work that we have done till now, and we understood the role of different host factors. Just to name those, I have put all these our publications on host proteins, where we have seen the role of heat shock protein, and definitely this MAP kinase pathways, and as I mentioned that 81, and these are few other molecules, that IRGM, and then trip channels. Also, it is very interesting that we have seen that DNA damage response. You think like why these viruses are even damaging DNA of the host cells, and this being RNA virus, what it is doing by inducing DNA damage response of the host cells. So what we have found that virus actually hijacks the different host pathways and then utilize these machineries for its own use. So this was interesting to know, because usually these are not much common event for RNA viruses. And what we have seen that the viruses are very clever. It induce the DNA damage response, so that now the host will synthesize all the repair pathways, and then the virus can hijack this.
Ma'am, if you could please come.
So I am at the
Yeah.
end. So I like to acknowledge all my collaborators. There are a huge amount of people who are involved in different works with my group, and definitely Dr. Shubhuti from Shua University, Dr. Parida from DRD who gave me the virus, and Dr. Shubhashi Chattopadhyay for all the immunological work. And this is the people who are working with me. Definitely the funding agency. I have grants from CSIR, DST, BIRAC, and other funding agencies too, DBT. And thanks a lot for your kind patience here.
I would like to thank Dr. Soma Chattopadhyay for her exceptional talk. We'll take only one question because of time constraint. Yes, Professor Kundu wants to ask some question.
Yeah, Dr. Soma, excellent talk. So my question to you, either your TM inhibitor or that second set of inhibitor, when you are giving to these infected mice, other than macrophage, or even within the macrophage, are they targeting M1 or M2, or whether they are targeting other immune cells other than macrophage? And the second question, when you are talking